So deriving information from speech is one of the most critical functions of the human auditory system. It's usually pretty effortless, but as we all know, speech perception in actuality is a highly computationally challenging process. And that's due in part because factors extrinsic to the speech signal substantially alter the acoustics. And this includes environmental factors like imperfect sound transmission, reverberation and echoes, and the presence of background sounds, all of which alter the acoustic waveform that reaches the ear in different ways, but they leave the perceptually relevant information like the words being said or the talker identity unchanged. So in order for successful communication to take place, the auditory system needs to represent speech in a way that is invariant to or tolerant of these types of acoustic variation. So this invariance problem is a fundamental challenge of all perceptual systems, and invariant object recognition has been studied for decades in the visual system and is thought to be achieved by hierarchical anatomical computations that increase invariance across anatomical brain regions, as well as hierarchical temporal computations that increase invariance over time within a region, in part reflecting recurrent connections. By comparison, much less is known about the neural computations that underlie invariant representations of speech in the human auditory cortex. And that's because much of the relevant research has been conducted in non-human animals that lack the speech-specific computations that are present in human non-primary areas. And also much of the previous human work has used non-invasive neuroimaging methods that don't have the spatiotemporal precision necessary to measure the time course of invariance to speech, of speech responses. So in an attempt to bridge these gaps, our lab's research examines invariance in auditory cortex using spatiotemporally precise intracranial recordings from the human brain. And for this project, we collected data from 14 neurosurgical patients at the University of Rochester and the University of Iowa, who had a total of almost 2,000 implanted electrodes, 167 of which were located in and around auditory cortex and responded to sound. And in order to quantify invariance, we measure the intracranial responses to a large number of words. Here you can see spoken words. Here you can see the waveforms of 3 example words and their corresponding high gamma responses recorded from a single electrode. And then we also record responses to those same words presented with different types of acoustic variation, so like reverb, spectral filtering, and background sounds. And then we quantify the extent to which the response is similar across these two conditions by correlating the neural responses to the words with and without variation separately for every time lag relative to word onset. And we call this the invariant correlation, or IC, and it provides a time-varying measure of the strength of invariance of a given electrode's response. However, even if a response is perfectly invariant, because of the neural signal is noisy, the response is never going to be identical in these two conditions. So to account for this, we compute a noise ceiling measure for our invariant correlation by performing the same correlation between 2 repetitions of physically identical stimuli. And in practice, we don't present these words in isolation. We present them in 2 different orders, so natural sentences and then words shuffled in a random order. And because of that, the preceding context before our words is independent in the 2 conditions being compared for our measures, meaning that the correlations will be 0 before word onset, and this allows us to quantify the buildup of invariance over time. And just to build intuition with these measures, so if there's no shared representation between clean speech and speech with these added variations, the IC will be 0. On the other hand, if an electrode's response is fully invariant to these types of modifications, the electrode response will be the same to a given word regardless of whether or not there's this added variation, and the IC and CC will be the same. So now I'll walk through 3 hypotheses that we tested as to how invariant speech representations might arise in auditory cortex, and keep in mind that these are not mutually exclusive. So first, we tested whether neural populations show strong invariance that generalizes across our 3 very different types of variation, or whether different neural populations specialize in generating invariance to different kinds of variation. Second, based on what's known in vision, we hypothesized that we would observe a hierarchy across anatomical regions such that the further away from primary auditory cortex you go, the more invariant responses become. And an intriguing variant of this hypothesis is that invariant speech representations might emerge only in non-primary areas that are speech selective. And third, we tested for the existence of hierarchical temporal computations, which would manifest as an increase in invariance over time, such that the IC starts out below the CC and then reaches the CC at some point after word onset. So we computed these measures, our IC and CC, for each electrode individually, and then across electrodes we observed really substantial differences in the strength and timing of invariance. So for some electrodes, like the 2 I'm showing here, adding acoustic variation caused the invariant correlation to drop relative to the CC, indicating that they're somewhat sensitive to these types of acoustic variation. Whereas other electrodes showed near complete invariance with the IC equaling the CC right from the get-go. So this electrode responds pretty much the same, regardless of these dramatic environmental variations. And interestingly, we found that for the most part, the strong invariance in these electrodes generalized across the three very different types of acoustic variation. We also observed other electrodes with responses that became more invariant over time. So for example, in this electrode, the IC was initially lower than CC, but by about 200 milliseconds after word onset, the IC reaches the CC and the response becomes fully invariant. So this is consistent with this hierarchy across time, in which an initial non-invariant representation is refined, potentially through recurrence. All right, so I've shown some examples of what we see, and next we sought to see whether we could explain these patterns of invariance across auditory cortex. So one hypothesis that I alluded to is that speech-selective neural populations are involved in invariant coding of speech in the human brain. So to test this, we ran a separate experiment in these same patients where we measured cortical responses to a large set of natural sounds from all different kinds of categories, and then we used that data to measure the speech selectivity of each electrode. And we chose subsets of electrodes that were maximally speech-selective or non-selective. We then measured the invariance of these groups of electrodes, and what we found was that on average, speech-selective electrodes showed stronger invariance than non-selective electrodes. And moreover, in speech-selective electrodes, the strength of invariance increased over time, with the IC reaching the CC again about 200 milliseconds after the onset of speech information. So importantly, we ran some additional analyses, and we verified that the strength of invariance of these speech-selective neural populations goes above and beyond what you would expect given their hierarchical position in non-primary auditory cortex. And we also ruled out the possibility that the effect would be explained by things like spectrotemporal modulation tuning. So to summarize, we developed a new paradigm for quantifying the invariance of neural responses, and using this paradigm, we're able to uncover neural populations whose responses to speech are nearly completely unaltered by many distinct types of extrinsic variation, like reverb, background sounds, spectral filtering. And these neural populations are clustered in speech-selective regions of non-primary areas and rapidly increase their invariance over time, showing strong invariance approximately 200 milliseconds after the onset of information. So there's a lot more that I didn't have time to get into here, including additional control analyses and a computational model that we developed to predict invariance in individual electrodes. So come to poster 45 after lunch to learn more. And I'd like to thank my PI, Sam Norman-Hagner, our lab, our collaborators at Rochester and Iowa, and our funding sources. Thank you.
Okay, thank you very much for this wonderful talk and fascinating data. We have time for one short question. Yes.
Thank you, Dana, for this really interesting talk. What were your participants doing? And do you think that affects what the degree of invariance that you see?
Yeah, so these are epilepsy patients, so we wanted to make sure that they were, engaged in the task. So they were listening to our stimuli and listening for a target word, one of the words at the beginning of one of the sentences, and then pushing a button. I would guess that it's important that they're paying attention to the speech, but yeah, I can't really speculate beyond the importance of sort of perceiving and understanding the speech.
Thanks.
Yeah.
All right. Thank you very much.
Great.
Then let's thank again Dana.